All right, good evening, everyone. It's great to see everybody here. This is the last Medical Scholar Seminar of this semester. So just as a reminder, for those of you who are new to this series, this is the seminar series that focuses on physicians who are actively doing research and caring for patients. And we feel that this is an important series because it does allow us to sort of emphasize the importance of observations that come out of the clinic environment and really inform that discovery process. So tonight we're excited to be able to share with you a speaker who is coming at the invitation of one of our very own, Dr. Nick Ryder, and he's going to give us a brief introduction.
Good evening, everyone. So Dr. Keller would hope this would be a little bit more brief than it is, but it is a true pleasure to introduce my friend and colleague, Dr. Michael Keller. Dr. Keller is an associate professor of pediatrics at the George Washington School of Medicine. He's also an attending physician in the Division of Allergy and Immunology at the Children's National Hospital in Washington, D.C. He's the director of the Translational Research Lab in the Program for Cellular Enhancement Technologies for Immunotherapy and the Center for Cancer and Immunology Research at Children's National as well. Dr. Keller completed his undergraduate degree in 3 years. What a slacker. At the University of Virginia, followed by medical school at Duke, where he graduated AOA. He then went on to complete pediatric residency at the Johns Hopkins Hospital, followed by an allergy immunology fellowship at the Children's Hospital of Philadelphia. He's authored over 70 peer-reviewed publications in addition to over 20 book chapters, review articles, and editorials. He's known for his international excellence in patient care as well, loved in our community for his patient care abilities, and has been a mentor to very many students, undergraduate students, medical students, graduate students, and postdocs. His research foci impinge on cell-based therapeutics, and in particular, adoptive immunotherapy for the treatment of viral infections in patients with primary immune disease. He's won a number of awards. I won't list them here, but I'll just say a number of international and national abstract awards for presentations. And if that wasn't enough, he's an avid outdoorsman and has plans to climb Mount Rainier this summer. The title of his talk is Immune Special Forces, Virus-Specific T Cells for Combating Infectious Diseases in Immunocompromised Patients. Please join me in welcoming Dr. Keller.
All right. Thank you very much, Dr. Ryder, and huge thank you to faculty at Virginia Tech and Carilion for the invitation to come and speak with you today. So here are my disclosures, and I do want to importantly get out of the way the elephant in the room. So I did go to UVA, and I did go to Duke, and my wife went to UNC and University of Maryland. So I'd like to think of myself as, in order to keep peace in my house, a neutral observer of the ACC. So here's a quick overview of a little bit of background about my clinical focus and about adoptive immunotherapy, talk about a case, and then talk about my lab's role in trying to advance cellular immunotherapies in the infectious disease space. So first, as I'm a clinical, like Dr. Ryder, I'm a clinical immunologist, and since we're going to be talking a lot about inborn errors of immunity, I first wanted to at least define this quickly. So to date, there's over 500 genes that have been tied to various forms of inborn errors of immunity. They're also known as primary immunodeficiency disorders, which are rare diseases that impact the function or development of the immune system. These can manifest in many different ways, as shown in this Venn diagram on the right, including autoimmunity, severe atopy, cancer risk. But the most common manifestation is infections. And the Jeffrey Modell Foundation published the 10 warning signs of primary immunodeficiency, which focus heavily on incidence and severity of infection as red flags for inborn errors of immunity. T cells are you think of as the generals of the immune system and are critical for mounting sterilizing immune responses as well as establishing immune memory. And some of the most severe forms of inborn errors of immunity, including severe combined immune deficiency, which is also colloquially known as bubble boy disease, and related disorders, compromised T cell immunity. And this middle graph here shows David Vedder, who is the media known as the Bubble Boy, who survived 12 years at Baylor College of Medicine. And that's actually Bill Shearer, Nick's former boss at Baylor, who was David's physician, and survived in a sterile environment designed by NASA. Newborn screening has been implemented in 50 states as well as several other countries to identify children with SCID and related diseases and to allow us to establish curative therapies. Patients undergoing cancer chemotherapy and hematopoietic stem cell transplant are similarly at profound risk of severe viral infections due to low T cell immunity. And as in the lower right is showing, for instance, a patient with CMV pneumonitis, which we see sometimes, unfortunately, in patients with compromised T cell immunity. So I'll start by discussing one of our patients, and this is a boy with severe combined immune deficiency that was identified in the DC region by newborn screening, found to have IL-7 receptor deficiency, and underwent a bone marrow transplant at 6 months of age on a clinical trial testing a antibody-only conditioning called anti-c-Kit, but unfortunately had primary engraftment failure. He was retransplanted from his mother at 12 months of age, but prior to that had developed cytomegalovirus viremia, which recurred with pretty severely early post second transplant in spite of multiple antivirals. So here I'm showing you his clinical course in setting of that second transplant, and the viral load is in red here. And you see, so he had fairly significant viremia in spite of being on multiple antivirals, ended up in our intensive care unit with respiratory symptoms, with high CMV viral load and respiratory samples and presumptive CMV pneumonitis. Respiratory symptoms improved with adding letermovir, but his viral load continued to climb in spite of these antivirals. So this case highlights a typical scenario in management of infections in immunocompromised patients, including those undergoing hematopoietic stem cell transplant. In most of these transplant patients, we worry most about bacterial infections early prior to neutrophil engraftment, but viral susceptibility persists until T cell constitution, which can vary from anywhere from 6 to 18 months post-transplant. And while new methods of mismatch transplantation, such as using ex vivo T cell depletion, have substantially improved our donor pool and our overall outcomes in transplant, they come with a prolonged risk of viral infections. Occurrence of graft-versus-host disease and need for prolonged immunosuppression also impacts this viral risk. And data from the CIBMTR, which is a national data collection group out of Minnesota, it mainly focuses on bone marrow transplant, demonstrated that infections shown here in gray still account for up to a quarter of transplant-related mortality, both in adults, as shown on the lower row here, and pediatrics in the top row, and especially in the first 100 days post-transplant. A recent study in Australia and other studies demonstrated that viruses are the leading cause of infection-related mortality in children, with adenovirus and cytomegalovirus being the most commonly isolated viruses. Antivirals are the standard of care for prevention and treatment of viral infections in hematopoietic stem cell transplants. Treatments for CMV are decent, particularly with the addition of letermovir prophylaxis, which is a terminase complex inhibitor. Options for adenovirus and BK virus are still very limited, with tenuous efficacy with cidofovir, which is a chain terminator. Additionally, toxicity is a major hurdle for many of these drugs. Marrow suppression is a major concern, particularly with polymerase inhibitors like ganciclovir. Renal toxicities are a big limitation with broscarnate and cidofovir, and GI toxicities can also occur with maribavir and letermovir. Resistance is also a growing limitation. Up to 30% of CMV isolates can harbor resistant mutations, including actually mutations in the terminase complex for letermovir, which is really the newest member of our armamentarium. In patients with inborn errors of immunity like SCID, presence of active infections is well described to worsen survival rates for bone marrow transplantation. And most of these infections in patients with SCID are due to respiratory viruses or DNA viruses, as demonstrated by our previous natural history studies. In a recent paper published in The Lancet, the Primary Immunodeficiency Treatment Consortium showed that even in the current era with newborn screening and modern antivirals, active infections worsen survival, as shown here in this multivariate analysis. Several studies have also tied T cell recovery to survival after hematopoietic stem cell transplant. And this study from Russia authors analyzed total T cell count at day 30 post-hematopoietic stem cell transplant and found that for patients with less than 1 T cell per microliter of blood, there was a substantial increase in transcellulated mortality, as shown here between the green curve and the blue and purple. And in these studies, non-relapse mortality was predominantly due to viral infections. For over 30 years now, investigators have been studying the use of T-cell immunotherapy to try to restore antiviral immunity in immunocompromised patients, including those after bone marrow transplant. Stan Riddell and colleagues at Hutch in Seattle first demonstrated this method by growing CMV-specific T cells with infected skin fibroblasts from bone marrow transplant donors, which are then given to recipients post-transplant to alleviate the risk of CMV. Helen Heslop and team at Baylor used EBV-infected lymphoblastoid cells to produce EBV-specific T cells to combat EBV-associated lymphoproliferative disease, which are given to over 200 patients over a roughly 10-year period. Their team later developed a chimeric adenoviral vector that expressed the CMV PP65 antigen, which when infected into EBV lymphoblastoid cells as antigen-presenting cells allowed the production of T cells that simultaneously targeted CMV, EBV, and adenovirus in one culture. And these were given into patients in several subsequent phase 1 and phase 2 trials. Most recently, Ulrike Gerdemann and colleagues also at Baylor showed that using synthetic viral peptide libraries could be used to rapidly produce virus-specific T cells without any live virus required. So when we talk about production of virus-specific T cells, these can be achieved either by selection or expansion methods. In selection, T cells are enriched based on either cytokine capture or multimer sorting, which has the advantage of using widely available platforms such as the Prodigy system and which have a quick one-day production time. However, the majority of studies have utilized ex vivo expansion, similar to what I described. In this classic method, T cells are stimulated over several weeks with antigen-presenting cells that display viral antigens. This is further simplified using the rapid expansion method from Gerdemann and co. In which PBMCs are pulsed with overlapping viral peptides and then cultured for 10 to 12 days with cytokines and bioreactors. Following ex vivo expansion, we see a predominance of effector memory cells with both CD4 and CD8 populations, and the proportions of which usually depend on the targeted virus. So here I'm showing you on the left single cell transcriptional profiles of 4 of these products that were concatenated. We again see a mix of CD4 and CD8 populations with evidence of activation and proliferation based on the gene expression profiles. We also confirm this by flow cytometry on one of these products, which again shows effector memory populations based on CD45RO expression in both the CD4 on the lower row and CD8 populations, as you see here. And in another view of one of our VST products, following CMV peptide restimulation, we evaluated by a 38-color flow cytometry, spectral flow cytometry panel, which we then analyzed by t-SNE plots, as shown here on the left. This was done in collaboration with our flow cytometry core, Cliff Zarsky and Gage Paul, as shown here. So we see 2 dominant populations. And we see multi-cytokine expressing CD4 cells, as shown on the top, and let's say here in blue, and then memory CD8 cells on the yellow on the bottom. If we gain a latter population, and then we can evaluate activation and co-inhibitory receptors in either CMV or actin restimulation. And we see that following 12 days of culture, there's no appreciable increase in co-inhibitory receptors, suggesting that in short term, we're likely not inducing T cell exhaustion. So during VST production, we can also alter the final product by modifying the cytokine cocktail used during expansion. Here I'm showing data from several cytokine combinations we tested for production of CMV-specific T cells. The top graph shows total T cell expansion, and the bottom is interferon gamma expression following restimulation. IL-4 and 7 is our legacy cocktail, which most commonly used in our production. That's in purple. IL-15 and 6 is in green, and IL-15 alone in red. And what we saw was that 15 and 6 in combination seemed to give us the best boost, particularly for CD8 expansion. And then all 3, in terms of interferon gamma production, really maintain that population roughly equally. So we do this generally by viral target in each time to kind of really hone in on the best combination that gives us the T cell population that we want. So moving on to a clinical application, in our experience, VSTs have been a really helpful adjunctive therapy, particularly for high-risk patients. And one of our early high-risk patients was a child with Wiskott-Aldrich syndrome who developed EBV reactivation that did not respond to rituximab. We gave a single dose of virus T cells from his bone marrow donor. And then as you see here, the virus shown in red dropped off pretty nicely. And we saw then as virus dropped off, an increase in T cells in his peripheral blood that recognize dominant EBV antigens LMP2 and EBNA1. And generally inversely correlates with the viral load in the weeks after infusion. VSTs were first utilized, as I mentioned, mostly in the bone marrow transplant setting. And I'm showing here some combined data from pretty much most of the studies of the last 20 years. So BMT is the most common application, but however, that use is expanded to treatment of viral infections in recipients of solid organ transplantation, patients with HIV, and even in patients with inborn errors of immunity. CMV remains the most common target, followed by Epstein-Barr virus, BK virus, adenovirus, and then new targets are also coming up, including SARS-CoV-2, JC virus for progressive multifocal leukoencephalopathy, and others, as we'll talk about. In terms of safety, VSTs are generally fairly well tolerated. When looking at prior studies in the bone marrow transplant setting, low-grade graft-versus-host disease generally occurs in roughly 8% of recipients, high-grade or chronic GVHD in 6%, and comparing expanded versus selected cells, GVHD is also roughly equivalent, maybe slightly higher in selected cells, but again, fairly comparable to background rates in recipients of allogeneic bone marrow transplant. High-grade toxicities such as high-grade cytokine release syndrome, although very, very common in the setting of CAR T therapy, are luckily fairly uncommon in recipients of virus T cells, although we do see it. It's generally only seen in the setting of very severe infections with high viral loads, as we'll talk about. So in comparing efficacy rates in solid organ transplant versus bone marrow transplant, and again, we're generally defining that by at least a sustained reduction, if not a resolution of viremia and attributable symptoms. We see fairly comparable rates of efficacy across prior studies. So here it's 78% in SOT versus 80% in HSCT. Efficacy in expanded versus selected cells also are fairly comparable in prior studies. Here I'm showing medians of 90% for ex vivo expanded versus 77% for selected. Albeit, again, these are all different populations, very heterogeneous, and most of them basically are non-controlled. So to briefly shift to our role in our immunotherapy program at Children's, we came there in 2013. We admittedly were an offshoot of Baylor, where our boss, Kath Ballard, had been for almost 20 years. And our goal was to develop a cell therapy and gene therapy center within the DMV. Within a year, we treated our first patient on study. This is a boy with primary deficiency who got donor-derived VSTs. Over the course of the next 5 years, we began several consortium studies, including through the Children's Oncology Group and the Pediatric Transplant and Cell Therapy Consortium, where we were able to partner with centers across the U.S. to enable these studies. Our GMP also got a pretty substantial upgrade in 2022, and then most recently, Kath and some of our team got a $25 million Cancer Grand Challenges grant supported by NCI and Cancer Research UK. And our recent roundup now is 300 patients treated over 12 years, 14 IND studies, 4 of which were first in human. And I'll now shift a little bit to with that prolonged background, I'll shift a little bit to what my lab really focuses on. And we focus mainly on expanding viruses that we can target, trying to work on improving efficacy through gene editing and other manners, and then enhancing use and accessibility. So first to talk about expanded targets. One of our earliest attempts at adding new viruses to our armamentarium was targeting human parainfluenza virus. So this is enveloped RNA virus that is both common and can be severe after bone marrow transplant that has no available antivirals or vaccines. We've previously demonstrated that nucleocapsid and matrix proteins were very strong T cell antigens. And in these prior studies, we were able to map T cell epitopes in the matrix protein, which is on the surface of parainfluenza 3, most of which were CV4 restricted and occurred in several hotspots in the protein, as shown in the lower left, including C-terminal domain, which is required for capsid production. Many of these epitopes notably are also shared with parainfluenza 1, and we were able to demonstrate cross-reactivity between these viruses at these epitope sites. So based on that, we opened a protocol for treatment of patients following a medical stem cell transplant with hexaviral VST, so targeting all 6 of these viruses. Our GMP director, a big baseball fan, helped lead us the NATS trial, which again, depending on the year, could be a good or bad thing. And that's run by Hannah Kinoshita, one of our junior faculty. So here I'm showing you 17 of the products that were made under the NATS trial for different patients. So here you see that specificity of viruses vary, which relates primarily to the donor's immunity to these viruses. So CMV in blue and EBV in orange, very prominent in donors who are seropositive, such as 2, 3, 7, 11, and 12. Whereas responses to adenovirus in gray and parainfluenza 3 in light green are fairly ubiquitous. Most people, luckily, are immune to these viruses. And overall, the VSTs that we made were recognized immunity of 6 of the 12 viral antigens that we included during expansion. So we generally were able to get immunity to most of these viruses. So to date, we treated 17 patients on the studies, 15 on the conventional, and then 2 via EIND. And 10 of them had active viral infections, and we got a clinical response in 9 of 10, including 4 or 5 with adenovirus, 5 or 6 with CMV. Antiviral responses generally correlate with detection of these antiviral T cell responses in the peripheral blood. So in the upper right, I'm showing you one patient who received VSTs for treatment of CMV. Sorry, the contrast is not great, but the CMV is this dotted line here. And it is, you see the viral load on here. So it's not astronomical, but it was enough that definitely required antivirals. And after receiving VSTs, had resolution of the viremia, and then in the weeks thereafter, we were able to see T cell responses in peripheral blood against CMV PP65, shown in orange. And when we looked at TCR sequencing, also from the peripheral blood, we saw an increase in clonotypes corresponding to the VST product that was given. And that also corresponded to the resolution of the virus. So generally, as the virus dropped off, we saw more of the repertoire that was specific for that was corresponded to the infusion given. The infusions are generally well tolerated. We did have 2 minor cases of CRS in this study, including so circling back to our SCID patient I mentioned earlier. So we again had pretty substantial CMV on multiple antivirals. We gave a 1 dose of VSTs derived from his mother, who was a second bone marrow transplant donor at 50 million cells per meter squared. You see he had ended up with a little bit of uptick in his respiratory symptoms, and bounced back to our ICU, got 1 dose of tocilizumab, and that resolved. So he was in and out of the PICU quickly, and then his viral load resolved. We were able to stop all antivirals, and he did great. And here he is, he's going 1 year out from transplant, doing wonderfully. So that is kind of the ideal scenario. So though virus-like T cells have been very useful adjunctive therapy at our center, the degree of customization is really limiting. You have to have an available donor. For most of these trials, that donor has to be immune to the targets of interest. This leaves out those undergoing gene therapy, cord blood transplant. There's also the time requirement for VST production release testing. So again, not ideal for someone who's critically ill. So instead, if we use banked VSTs from healthy donors, pre-made, well-characterized, we can circumvent these limitations. So third party, as we term it, VSTs allow an off-the-shelf salvage therapy, whereas if a patient develops a viral infection that's poorly responsive to therapy or undue toxicities, we can turn to our bank, find a partially HLA matched product that can be given. Clinical responses can be obtained even with a low degree of HLA match, as long as we are sure that antiviral activity is known to be restricted through 1 or more of those shared HLA alleles. And these cells are able to expand in vivo. They're generally only short-lived. Once these patients reconstitute, those cells are generally kicked out, which we'll talk about in a bit. But even at low cell volumes, generally in the range of 10 to the 7th to 10 to the 8th cells per meter squared, we can often see responses within a month of infusion. And this approach to what was termed third-party VST therapy was first pioneered by Dorothy Crawford and colleagues in Scotland, who use EBV-specific T cells to treat drug-resistant PTLD. Baylor College of Medicine, Malcolm Brenner, Helen Heslop and co. Did the first multi-virus trials, achieving 74% response rate in their first study, 92% in their second, targeting 5 viruses. Our colleagues across the world, the Westmead Institute in Sydney and Cincinnati Children's, Sloan Kettering, and we have also run successful third-party trials. And all these trials, rates of graft-versus-host disease are surprisingly low and generally low grade. So in our recently completed ACES study, where we treated 51 patients at 22 centers across the US, we saw an overall response rate of 65%. I stress this is a real world study in that we treated patients regardless of how severe they were, including several in the ICU. We had an overall significant improvement in overall survival in patients that did have an antiviral response, as shown in the lower left. In some cases, those responses were fairly dramatic. We had a 19-year-old with a rare disorder called X-linked lymphoproliferative disease type 1, who had EBV post-transplant lymphoproliferative disease after transplant. So here's EBV viral load is in red here. He got rituximab first. His viral load went down, but he still had disease. So on PET-CT at day 127, he still had nodal disease in the cervical, axillary, and hilar lymph nodes, as shown in this PET scan here. We gave 1 dose of a third-party VST at day 147. And then on re-imaging by PET at 203, complete resolution. He needed no further therapy. And you'll see pictures of him in a little bit, but he went on to play football for Michigan. Luckily not ACC. Notably, so though you obviously need a partial HLA match for VST therapy to be effective, the degree of HLA match does not seem to correlate in our prior studies with chance of response. So in our first studies at Children's, look at overall match, as well as high resolution class 1 and class 2 match in third-party VST protocols, found no difference in the spread of responders or non-responders in terms of their degree of HLA match. And this, again, for class 2, this made sense to us. There's a fair bit actually of promiscuity between HLA classes, which again, we can talk about a little later on. So in evaluating immunoconstitution, as performed by our technician, Nassim Agazian at Children's National, we showed that after third-party T-cell therapy, that most patients had a detectable increase in the magnitude of our specific T-cell immunity in the peripheral blood. So on the left here, these are interferon gamma ELISpot results against CMV and adenovirus before infusion, and then at the post-infusion time points, generally at days 28 to 45. And the uptick in immunity was significant for adenovirus. And by flow cytometry, we can also see this increase in antiviral immunity based on cytokine expression. So here you see this is a TNF alpha and interferon gamma on the axes in the CD4 populations, CD8 populations for CMV, and CD4 and CD8 for adenovirus. And you see that at day 28, we saw pretty good T-cell population, CD4 population for adenovirus, stronger CD8 populations for CMV, which is pretty typical for these two viruses. And then most recently, we've looked at patient blood before and after third-party infusion by single-cell RNA-seq. This is a collaboration with one of our collaborators, Chintan Parikh from City of Hope in California. So this is composite results from several patients. Pre-infusion, you see that there's early stem cell memory, but really no effectors. And also, I'll point out up here, we saw some evidence of T-cell exhaustion based on the transcriptional profile. And looking again at roughly the 1 month time mark, and this is again several patients concatenated, we see a real ballooning of the memory effector populations, and we see resolution of that exhaustion profile. Another virus we've been working at target lately is norovirus, which is a leading cause of epidemic gastroenteritis, and in immunocompromised patients can cause chronic diarrhea and wasting that can last from months to years. There are no antivirals, no effective vaccines. There are 4 main genotypic groups, of which type G24 causes the majority of clinical outbreaks. And in collaboration with virologists at NIAID, we screened healthy donors for T-cell responses. We picked the Sydney 2012 norovirus G24 strain to make our target antigen libraries. And we saw that following expansion, the vast majority of healthy donors had response to 1 or more of these norovirus antigens, and that the major capsid protein VV1, shown in green here, and the protease NS6 seem to be very immunodominant. So based on that, we moved those on to a clinical trial, which I'll talk about in a moment. Predictably also, so antibody seropositivity correlated with T cell response, which was, again, not surprising, very consistent with what we see with most other viruses. We mapped the T cell responses in healthy donors and identified both CD4 and some CD8 restricted epitopes throughout the NS6 and VP1 proteins. In NS6, these included several epitopes surrounding the catalytic protease sites near the N-terminus. And in VP1, we saw clustering at the shell domain here, and also at the C-terminus in the P1 protruding domain. And since norovirus has many circulating strains that differ at several of these epitopes, we asked how cross-reactive these T cells were against mutated epitopes. Overall, we found that CD4 epitopes had a high degree of cross-reactivity, whereas CD8 epitopes were only moderately cross-reactive, which tracks with what we've seen in terms of promiscuity. Generally, class 1s, especially anchor peptide changes, are less well tolerated. So we made a epitope cross-reactivity index, generally saw between 40 to 60% cross-reactivity, which was reassuring to us that likely that if we produce these norovirus T cells, they should provide some protection against circulating strains. And recently we studied 2 patients with SCID who came to transplant with chronic norovirus. And here I'm showing you, it's a little complicated, but this is the clinical course for one of these patients. It was a 1-year-old with X-linked SCID who had norovirus, SARS-CoV-2, and disseminated BCG. So very, very sick young man. The norovirus load is here in red. So this is log scale copies per gram of stool. So very high going in. He had alemtuzumab only transplant because of the degree of infections. But in the months after transplant, we saw a drop in his stool shedding, really most notably concomitant with an increase in his CD4 count, shown in blue here. To a lesser extent, his CD8 count in green. And then when we looked at his norovirus immunity, we were able to get 2 time points, day 33 and day 209. We saw a notable increase in both CD4 and CD8 T cell populations targeting several of these norovirus proteins. So VP1 and a 6 and a CD4 compartment. And then in spite of the small CD8 compartment, we saw a pretty robust NS7 response. So again, this to us suggested that that restoration of T cell immunity to these viruses is likely important for clearance. So based on this work, we opened a phase 1 study of norovirus T cells, either in the setting of bone marrow transplant or in the setting of inborn immunity, solid organ transplant. And we recently are expanding to CAR T recipients. And we've had a surprising number of referrals of chronic neuro and CAR T recipients. We had 4 patients treated so far, one of whom cleared at NIH, we're happy to say. We were hoping that maybe Royal Caribbean or other cruise lines might line up to support this. Perhaps we could be part of like a platinum package. No love so far. No love so far. All right, moving on. So in terms of improving efficacy, one particular interest, I mean, we not uncommonly we see patients that we'll treat them with VSTs and have absolutely no impact. And in the ACES study, that third-party study that we completed, we looked by univariate analysis at clinical factors that impacted the chances of us obtaining a clinical antiviral response. And fairly predictably, we saw that use of corticosteroids, incidence of graft-versus-host disease, and use of other T cell agents like antithyroid globulin reduced our chances of response. Interestingly, also, a rare inflammatory complication called transplant-associated microangiopathy, or TMA, and treatment of it with eculizumab, an anti-C5a agent, also substantially reduced our chances. So that made us think actually that complement actually may have more of an impact on T cell survival than we previously realized. But this again convinced us that if we want to treat preventatively early, we're going to have to turn to gene editing. So alemtuzumab is a common monoclonal antibody that's used for lymphodepletion. It targets a protein called CD52. It's on the surface of all lymphocytes. It actually doesn't have any known role in the immune system, and has actually been knocked out successfully in CAR T therapy in prior trials without any major adverse events. So alemtuzumab, despite its value in preventing graft-versus-host disease, definitely puts patients at risk for viral infection. So it's been of interest to us in terms of gene editing approach. Here I'm showing you, unfortunately, we had a previous patient with MHC2 deficiency, a teenager that came to transplant, had a matched sibling donor had received Campath as part of their prep, had rip-roaring adenovirus, gave VSTs at 28 days, but unfortunately we made absolutely zero difference. And we found out after the fact that his Campath level was still cytolytic. So again, just kind of highlights that this antibody actually can stick around for quite a while after transplant. And so in order to allow earlier preventative therapy alongside alemtuzumab, we turned to gene editing to knock out CD52 from our expanded VSTs. And we attempted this just in the setting of our rapid expansion protocol, where we would expand initially for 7 days, do CRISPR-Cas9 editing at day 7, and then complete the culture and harvest at roughly day 10 to 12. And this is led by our junior faculty member, Susan Conway, and our tech, Daisy Ayala Gomez. So we tested several lead CRISPR single guard RNAs targeting CD52 and found that, so here, this is a histogram looking at CD52, the first guide in blue, second in green. We found that many of them had very good knockdown efficiency, which we also confirmed by ICE analysis. We further demonstrated that in CD52 knockout VSTs, anti-CMV activity remains strong. This is upper right is showing LE spot against the CMV PP65 in green. So the first column is unedited cells here, and then the two lead guides are these two. So again, the antiviral activity is fairly intact. We showed that also by flow cytometry, the P65 cytokine expression is here. And then the overall phenotype of these cells also, edited versus unedited, was fairly comparable with a mix of CD4 and CD8 cells, a good effector predominance as shown here. And we also further were interested in whether or not CD52 knockout might change these cells transcriptomically. So we sorted the CMV3 populations using HLA multimers from edited or non-edited cells for again for dominant CMV epitopes, and then performed a single cell RNA-seq using the 10x system. So on the right, I'm showing you a heat map for top signaling pathways, including notch signaling, mitotic checkpoints, interferon signaling, mTOR. And you see basically, and again, first column here, unedited, second is edited. Generally speaking, they looked very, very similar. Didn't seem like we were inducing any major changes transcriptomally. And our PC analysis of these aligned very nicely, which again was reassuring to us. Additionally, whenever doing gene editing, we have to be worried about off-target editing. And in collaboration with the laboratory of Don Cohen at UCLA, we looked at off-target editing of our lead single guide RNA using a combination of in silico predictive analysis and 2 in vitro targeting assays, change-seq and guide-seq. We found a narrow number of potential off-target sites, and then using a method called RAMP-seq on edited or unedited primary human T cells, we saw minimal off-target editing, which essentially tracked evenly between the edited and non-edited cells, and no hits near exons or near oncogenes, again, which was reassuring to us. So to prove that these cells persist in vivo, we also have moved on to a mouse model where we gave edited or unedited cells to NSG mice and then gave them either alemtuzumab or PBS. And we showed that the infused cells, when marked by DIR stain, are clearly still persistent in vivo. And then when we give alemtuzumab, we see that these edited cells persist nicely in the mouse spleen, as shown in the first 2 columns here, whereas unedited cells are fairly uniformly eliminated. So another class of medication we're very interested in, and a little more earlier in terms of study, is what's called Janus kinase or JAK inhibitors. These are medications that are highly effective for treatment of graft-versus-host disease, but have a very known high risk of viral infections, particularly when given at high doses and in patients who are otherwise immunocompromised. So our team members have demonstrated that ruxolitinib, one of the early class JAK inhibitors, is associated with a dose-dependent drop in proliferation and antiviral activity of CMV-specific T cells. So here I'm showing you this is total cell number during rapid expansion versus ruxolitinib dosing, and here is antiviral activity against CMV. Again, PP65 is in turquoise, IE1 in dark blue. You see basically, especially at higher levels, get kind of dose-dependent drop-off, which is, again, similar to what we see in clinic. We generally will see viral reactivations when we have to give high doses of these medications. And mutations, interestingly, in JAK kinases have been seen in the setting of other diseases, including those in which JAK inhibitors are used, and particularly mutations in the genus homology, that JH1 domain, which contains the kinase activity, imparts resistance to ruxolitinib and to some extent other JAK inhibitors. And it's shown, and in several papers, these variants still permit downstream STAT5 phosphorylation, which is necessary for T cell activity, and permit cell survival in the presence of ruxolitinib. And then taking a cue from nature, we're now interested in modification of JAK kinases and VSTs in order to permit treatment of viruses while patients continue to receive these medications for inflammatory diseases. So another area of interest, shifting a little bit again, is the impact of the viral biology on VST therapy. Note we saw that in prior studies, a duration infection prior to therapy was significantly longer in non-responders than responders, as shown on the right. Baseline viral loads are often generally higher in non-responders, though this didn't meet significance in our prior population. However, we're interested in whether these patients with long-standing viral infections could have intrahost viral variation that could actually allow more common viral escape. So we've looked with a collaborator from UMass at one of our earlier patients with substantial CMV disease, for whom we gave 2 doses of VSTs and made absolutely no impact. And we did CMV genome sequencing was done in this patient with SCID over 4 time points. And what we found was 37 viral variants identified that met criteria for positive or negative selection, of which 7 were non-synonymous. No changes were seen in UL83 or UL123, which are the 2 viral genes that encode PP65 and IE1. But some variants were noted in viral evasive proteins that could potentially impact C-cell survival or function. So that's an area of interest that we're currently developing. So lastly, to focus, to shift to enhancing use and accessibility. We're very interested. So obviously, we were very interested in applications of VSTs, but this is a niche therapy. There's only 4 centers across the US that have this. And so one key question is which patients really truly need this? Who are the patients that are at highest risk? So in collaboration with the laboratory of Dr. Wei Li, who is now at UMaryland, and our grad student Pamela Chansky, we evaluated whether we could use machine learning to train an algorithm to predict the chances of clinical response in transplant patients who receive VST therapy. So Pamela used our recent clinical trial data, which she actually further expanded using a variational autoencoder. She then tested multiple methods to determine the most accurate method of predicting viral resolution based on clinical data. I found that the neural network approach seemed to get the best results. She went on to test this algorithm in 2 independent data sets from Cincinnati Children's and Westmead Institute in Sydney. And what we found was basically that we generally were getting F1 results, which again is a good typical, I'm sure Dr. Reddick can tell you at greater length about the computational details, but generally kind of a mark of accuracy and precision of generally about 70% with this algorithm, which was fairly comparable between the synthetic and real data. And we're hoping to extend this approach to further evaluate HLA matching, which presently is incredibly manual, and thereby improve, hopefully, matching donors and recipients for third-party VST therapy. So in summary, though T-cell therapy is very, very, very helpful adjunctive therapy, there's a lot of remaining questions and areas of study. To improve efficacy, we really need to be moving a lot further beyond just manual methods of matching. As the pandemic had highlighted, we really need to be far more aware of viral variation and epitope stability, as VSTs presently are primarily using static reference libraries for production. And then GVHD and other inflammatory diseases are pretty big hurdles and that require gene editing, which we're finally getting into. And we're hoping to bring our CD2 knockout product to hopefully a phase 1 trial within the next year. And lastly, to improve on safety, we really need better controlled trials of particularly of early T-cell add back, as most of our substantial adverse events tend to occur when treating patients who are very sick, very high viral loads, often in the ICU. And I suppose these are really probably missed opportunities to intervene earlier with a cell therapy product. So I'll close by focusing on the reason that I'm in the translational space. This is several more patients who receive VST therapy over the past decade. Some of them maybe would not necessarily be here if not for this therapy. And our patients' outcomes, really both good and bad, really press us to advance and improve this therapy. So that I'd like to thank Kath Butler, my mentor, our wonderful group at Children's, our many collaborators, and our funding agencies and foundations for support his work.
Thank you so much, Dr. Keller. We'll open the floor for questions.
Please.
There is a way to give the DSP
Yeah, that's a great question. Since we're really driven by our protocols, we generally, I mean, it's always, since many of these are either phase 1 or phase 2 studies. So it's usually a matter of kind of determining at what point are they eligible for our protocols. Generally speaking, I mean, if and when this is approved, my vote would be, again, if a patient is at high risk, if there's a T cell depleted transplant, if they have end organ disease, I mean, earlier is always better.
I have a question for you. Great stuff, lots of data. You mentioned during one of the portions that MHC matched and mismatched seem to have similar efficacy. Does that surprise you? If so, why? If not, why not? What's the mechanism that might explain that?
Yeah, that's a great question. And even in my field, it's a lot of debate about this. I mean, the hand waving argument is really that probably the quality of the match is far more important than just the quantity rather. And that really having at least one, if not more, shared HLAs that actually mediate antiviral T cell activity is probably more important than just kind of overall degree of match. For each of these antigens, interestingly, there's an immunologic hierarchy, which really differs by virus. It's really best studied for cytomegalovirus, but interestingly, some HLA molecules seem to mediate antiviral activity far better than others. Like HLA-B07 and AO2 are really classic ones for CMV, for instance. To the point where often if we do a mismatched transplant where a donor is AO2 positive and CMV positive and the recipient is not, a lot of that AO2 response from the donor, even if it's not it's not a T cell depleted transplant, will really not help the recipient just because of that mismatch.
Just for the folks who are online to be able to hear the question. Awesome talk, Mike. Really enjoyed it. Incredible work. And obviously you're serving our patients who otherwise, as you've shown, wouldn't have an opportunity for survival in many cases. And you showed a lot of, I think, really compelling data around the factors that relate to the host and even the virus. So kind of in keeping with the special forces theme, I want to ask you about super soldiers. Have you identified any factors in donors that actually allow for virus-specific T cells to be more potent than the average bear, if you will? And have you sequenced any of these folks that might like clear virus as a donor more effectively? Just curious if you have any insight into that.
That's a great question. Thank you. We really haven't, and we probably need to do that. Frankly, we do. I mean, there's data in the BMT literature certainly suggesting that younger donors are always going to be better. We actually following an adverse one pretty striking adverse event in our third party studies where we had a multiparous mom who in hindsight turned out to be actually HLA sensitized against a certain HLA. We have actually dialed back our recruiting to only young adults generally, and only women who have only had one or less pregnancies. But no, I think that's something we need to do, honestly, is try to understand who are really the ideal donors. I think early in our studies was just kind of whoever we could get in terms of volunteers.
Yeah, no, thank you. Amazing.
I have a question. What about for like the patient you had that had chronic norovirus going into, would that be a patient that you might want to do more of a custom instead of an off-the-shelf type of situation because you have more time?
Yes, that's a great question. And the kicker of we've been trying to kind of find those ideal patients for that Atlantic study for some time. And we've actually only used third party because we simply haven't, it's been, with the exception of that patient I showed, those 2 patients we published on, and that was early in the pandemic before Atlantic was open, unfortunately. So we couldn't actually make, it would have been ideal to make a custom product for them. So if we find none of those patients, absolutely, I would much rather use a donor-derived NST product in that setting. But to date, we've only, the 4 that I mentioned have all gotten third party cells just because none of them have been transplant recipients. But no, in every sense of the word, if we have the resource and the time, I'd always rather use a donor-derived product than a third-party product. Because again, third party is, it's only there transiently. As soon as they start immune reconstitution, it's gone. It generally is an okay bridge, but it's, I mean, if they get exposed again, it's not there. It's donor-derived on the hand is detectable. We found it's detectable for 10 years out. It just becomes part of their background repertoire.
Hi, thank you very much for the talk. There were some patients who received upwards of 3 doses of the VST infusion, and sometimes they received the same VSTs, and sometimes they received different VSTs. In some cases, they received 2 of the same VSTs, and then the third case, they received a different one. So I was just curious about what the rationale was behind switching up the donors, and was it just you're trying to get a response in really sick kids?
Yeah, no, that's a great, and thank you for reading that so in depth. Short version, so the decision to change up the products when we're using third party really depends. We have a committee at Children's that basically kind of debate these cases, and it's usually a matter of kind of was there intervening kind of was there immunosuppression use? Was there some reason that the first dose, if they think the first dose didn't work from a from an extrinsic standpoint, or was it simply that the match was not there or the product is not ideal? So in the latter case, we would ideally change the product if we have another suitable product in our bank. And that bank for that study, we had 40 products. We ended up only actually using, I want to say 18 of them. So it's again reflective probably of the American population. We had some minorities, but generally a lot of people of European ancestry. But usually it's we would change up basically if we had reason to believe that that first product was just not the right match for them. And again, we're usually trying to use epitope mapping to pick. In some cases, sometimes if we don't have a good epitope we've identified, then it comes down to just best HLA match. But that's exactly where we need more computational biology to kind of, take this away from manual and actually, analyze the data.
All right, great. Thank you so much. We appreciate you speaking to us tonight.
Yeah, thank you. Thank you so much.